Yeah, so thanks, Anita and Alex, for this really great conversation today. I'm going to talk about culturing human milk on Mars. I founded 108 Labs about 8 years ago, self-funded experiment and scientific and creative curiosity. Really didn't think of it like a business until we had some success, actually culturing human milk. And since then, it's really been kind of a roller coaster. But really a long time ago, we started thinking about the energy efficiency of using cells for producing food and other materials. And I think on Earth, when the cost really matters, it matters much less on Mars. And so it's just a really exciting thought experiment to think about cell ag on Mars. So why, we're going to talk about this today? Because milk is special. So sustaining human life with single cell agriculture is going to be the focus of my talk today. So again, Shayne Guiliano, just a quick background on me. I started out as an organic chemist. I worked with Hoveda on his metathesis catalyst, daily chromatography and NMR. Spent a couple of years really doing that daily. Decided that I wanted to move into something more creative. So I had a second career working in video games and scientific software. My first patent was for properties, bugs, and drugs, aggregated antimicrobial data across the country. Had a chance to work on life-saving software at Hippocrates as well, and video games at Namco and Imanji that have been downloaded and enjoyed by millions of people. So I've got a sort of diverse background. I've been running independent projects as well for almost 20 years now. It was mostly software for the first 12 of those years. And then I moved into biotechnology, self-funded, 108 Labs. Still to date, we've never accepted investment. The focus at 108 Labs right now on a product basis is a product we call Therapeutic, whole human infant formula with secretory IgA. I consider myself a cellular agriculturalist, so I run 108 Labs now. That was founded by Layla Strickland, PhD, and me in 2013. I also have a drug company called Therapeutics that's still in stealth, that is developing therapeutics using 108 Labs technology. And so let's just talk quickly about mammary cell bioreactors and what they can do and bring to the world. So, we have mammary cells, we have 3D bioreactors. Mammary cells really like 3D bioreactors. And what do you get? You get milk. So what is milk? I actually don't like to call what I make milk. I think milk comes from an organism. I call it milk molecules. These cells, I didn't appreciate what these cells make until I made them, honestly. But it is a who's who of important human molecules that have really struggled to be produced in any other system, fermented or any, they can have the same amino acid sequence, but that is not the same molecule. This is a 300 million year project that evolution has undertaken. And it's really the first drug that keeps us all alive. And humans have not even come close to approaching the efficacy and therapeutic benefit of human milk in any other food substance or drug substance. And human milk is evolution's Soylent Green, and that's really kind of the point. A single substance known to sustain human life. It's really the only substance that could make a claim to this. It sustains some babies for the first few years of their lives, actually, in some cultures that are exclusively breastfed. So why cell culture human milk on Mars? It's much easier to ship cells than milk or cows. Human cells can make human milk, and human milk can sustain human life. It's really that simple. There's really nothing else out there that you could make these arguments for. And so, I think that human milk has to be a part of the picture of Mars. But why human and not bovine? So, human milk is a little bit controversial on Earth, but bovine milk cannot sustain human life. If you feed a baby exclusively bovine milk that is not formulated properly, there's not enough tryptophan and it can die. So there is this single substance that was evolved, by humans for humans, and it's really, really special. Unpasteurized human milk molecules are fully nutritional, broadly antimicrobial, and beneficially probiotic. That is everything that you need to survive and thrive. Astronauts can use their own cells, and that's a really interesting sort of idea that on Earth wouldn't make a lot of sense, but on Mars, if in a pinch, male and female have these cells inside them. So you don't even need to necessarily ship your cells in liquid nitrogen to Mars. You can just extract them. The other point is that this cells made with human mammary cells are non-recombinant. These are actually, I believe we're producing the first non-recombinant proteins with biotechnology ever, and they're whole human, which I've defined as, human molecules made in human cells, human post-translational modifications, biosimilar to what you see in human milk. And that's really kind of critical for several of these molecules, actually. The lysozyme, osteopontin, some of these molecules that do not have the same bioactivity when they're produced in any other cell, whether it's CHO or algae or anything else, yeast. So it's kind of an interesting thought. Is drinking one's milk cannibalism? It maybe. I think it's a little bit of a controversial concept on Earth. On Mars, it just doesn't matter. It's all about survival, right? So in a survival environment, the import of single cell agriculture is much more obvious. We've never had enough human milk, so there's never really been an opportunity to even think about using human milk beyond the use of keeping babies alive. There's not even enough for the babies. We're losing about 3,500 babies a day around the world because there's not enough human milk. So it's a unique opportunity to think about human milk and the impact on human health. And it just isn't ethical to use this on Earth for anybody other than babies. But why milk? Milk is actually more than a food. If you pasteurize it, it's mostly a food. But if you don't pasteurize it, it's much more than a food. It's really a therapeutic substance. And in particular, it can have a big impact on the microbiome, more so than probably any other product. The microbiome, it's a hot field. Everybody's talking about it. What is the probiotic that we need to use to reset the microbiome to even approach stool transplants? And nobody's close. On Mars, it's even more important. Why? Because Mars is supposedly sterile. And maintaining healthy microbiome will be critical and difficult. We totally take for granted that there's more genes being, producing proteins in our body that come from our microbiome than come from our cells. So there's really nothing else known to support microbiome like human milk. So I actually think it's going to be absolutely essential for human milk, biosynthetic human milk, to be produced on Mars to maintain longevity of human health. Antibiotics may actually be quite dangerous on Mars because they are, very destructive to your microbiome. On Earth, it's fine. You just have to interact with the world to reconstitute your microbiome, and even then, you can, struggle to get back to a healthy microbiome often. But again, a total loss of microbiome could be very dangerous on a supposedly sterile world lacking probiotic microbiota. So again, milk creates our microbiomes now. Milk evolved for the specific purpose to initiate and support microbiome health with antimicrobial proteins to suppress the bad bugs and probiotic HMO and secretory IgA to support the good bugs. And this is completely overlooked. It's sort of taken for granted, really. Again, you see major health problems. Nutritionally, I think bovine does a great job, but you see really serious health problems that can emerge due to pathogen infection in populations that don't have access to human milk. No drug substance has ever approached mother's milk therapeutic effectiveness and safety in babies. Preterm infants, around 7% of preterm infants pre-32 weeks will succumb to some adverse outcome that requires extremely expensive interventions and can have a huge impact on the life of that baby for the rest of their life. When a mother breastfeeds, that same population, it's 1% adverse outcomes. So there is just nothing ever, there's no approved drugs for preterm infants today. And the standard of care is exclusive human milk diet, because these molecules are special. There's nothing that compares to them anywhere else. And they save lives now for our most vulnerable population. And again, human milk is unexplored for non-infants for the most part. There's no white market, if you will, of human milk. There are black markets, and there's some, niche sort of hidden markets of human milk that you could look at and kind of guess into some of the health benefits, which everybody who does participate in that talks about. But it's unethical, because again, the babies really need it, and there's not enough. So we've never really had an opportunity to explore how human milk can impact, non-infant health. But I think it's going to be essential. So how to make milk on Mars? So sort of instead of why, like how do we do it? Again, this is classic cell biology. There's no genetic engineering. I've never, we have some genetic engineering, but in terms of my cells, I've worked with some immortal cell lines, but it's primary cells for the most part. And the cell biology has really kind of been understood for decades. So we have, let me just struggling to get this moved. All right, so we have mammary cells. So the ingredients are basal media, growth factors, bioreactors, bioprocessing. That's it. This is pretty straightforward, and you get milk. Now thinking about that on Mars, how do we build a human milk industry on Mars? Do you ship or do you grow? So we have ingredients. What do we ship? What do we grow? What do we make there? I think you do both. I think probably forever. You're going to need new parts. You're going to, maybe have a crop failure on Mars. So there's going to have to, you want to build a closed system, but you probably want to continue to support it, and with occasional inputs from Starship. So just to dive a little bit into basal media production. So, this has, again, been worked out for decades. Chemically defined media, water, amino acids, small molecules, and media mixing and recycling. I was really, great I've thought a lot about recycling. I hadn't actually done a lot of research on it, because it's something that we don't think about a lot on Earth. Pascal earlier today was, talking about his research in this. And I think this is an underdeveloped science, really, to be able to feed ourselves with media and to understand what is needed in order to reconstitute that and get out any waste, that was produced, ammonia, lactic acid, whatever it might be. This is going to be much, much more important on Mars. And then, of course, the carbon and nitrogen sources. So we got to make glucose, we got to make glutamine. We need, and that's probably going to come from plants, I think. This is the mass that's consumed the most, and you need those atoms to produce all those proteins. And that's going to be, probably something that really has to happen on Mars. But there's a certain efficiency to thinking about milk production. The dry mass of, about 3/4 a liter of milk, the dry mass of media, if you add up all the mass of all the amino acids, the buffers, everything, sodium hydroxide, everything that you need. It's actually only about 40 grams can produce about 0.75 liters of milk, which I think, that's a daily productivity for one, decent sized bioreactor consuming about 2 liters of media per day. And that's not an outrageous number there. I think, you could imagine with a few shipments from the Elon Musk's rockets, that we can have enough raw materials on Mars to produce a steady supply of milk for a large number of colonialists. And so growth factors are trickier. There's different ways to approach it. So, I'm a chemist. I've done peptide chemistry in the lab. It's really painful. You get really low yields, but there are certain molecules, at least in the milk space, that you could probably make with peptide chemistry fairly efficiently. So there's some potential to think about that. I think plants are just going to really be important for production of raw materials in certain growth factors, where they're genetically engineered to produce the growth factors, or sources of amino acids. I think plants are going to be a big part of any closed system and industry for milk production. And of course, there's biotechnology. So there's E. coli and CHO are the typical platforms used to produce growth factors on Earth. I believe that the future of growth factor production is going to be whole human, meaning produced in human cells. This is a part of an area of my research, and I know there's other, this is something that's talked about often. I don't know what the final solution is going to be, but I think, we're going to have higher quality growth factors, if we move towards human production. But, certainly biotechnology is going to have a big impact on, reagent production for milk industry. And then, you're going to be shipping stable cell lines. You're going to be CRISPRing probably on Mars. You're going to want, if this fails, what is the next step? If this fails, if we have to start our own cell lines, we would always want to have, the ability to do that. But this is all achievable. I don't think there's not a lot of discoveries that need to be made, except maybe with, human growth factor production to get us to a point where we're, producing growth factors on Mars. And bioreactors. So, this is really where the biggest area of innovation needs to happen. It needs to be a modular closed system, I believe. And this is true for Earth and Mars. As we move towards food grade secretory production, at 108 Labs and other memory cell agriculture companies out there, this is the primary challenge for all of us. The FDA is just not going to let us do this. On Mars, you really want to, minimize the effort required to produce milk. So, those innovations are going to apply even more importantly on Mars. I don't think that there's too much about this that is unachievable. It's really just an issue of capital investment and bringing together, skilled pharmaceutical engineers and chemical engineers and process engineers to build the ideal closed system. But that's probably the biggest area of investment that we're going to need. Continuous production and validation. So you don't want to poison your colonialists. So there's going to have to be FDA-like commitment to the quality of our production and the consistency. Again, this is solved on Earth. I think it's achievable on Mars, but it's something that is going to require investment. And there's going to be challenges. Do sensors work the same? How do you deal with the different atmosphere? I mean, there's going to be a lot of challenges to producing a continuous production and validation on Mars. I think the ultimate goal, specifically with milk, again, if you take non-pasteurized milk and pasteurize it, last year I spent a lot of time focusing on COVID. And one of the most interesting pieces of data that I produced was pre-pandemic unpasteurized milk neutralizes COVID. So that is evolution's gift to mankind, that you don't even need neutralizing antibodies, because those molecules that I listed in the earlier slide are very, very special. We've never had a supply of them on Earth or Mars that didn't come from a mother, but these are very special molecules. And in order to, benefit from the antimicrobial, the nutritional acceleration, if, on Mars, calorie absorption, may be a little trickier, and there's enzymes in milk that, if you can maintain the bioactivity, can accelerate calorie absorption. And that's going to require, sterile closed systems from bioreactor to mouth. And then I think on-site manufacturing, we're going to have to make bioreactors on Mars. There's going to have to be a whole industry that is building these systems and scaling it up. I think probably everybody on Mars deserves that 0.75 liters of access to milk per day. I think, it's going to be important for us to maintain microbiome and just, overall health and that human milk can bring. So I wanted to just have a little creative slide here. I think, what should we call milk on Mars? I think, again, I don't like using the word milk that much because it's so controversial. On, if I don't know who follows this, I do, obviously, because I'm making it. But on Earth, there's with this recent debate: is it breast milk? Is it human milk? And it just doesn't matter. It's just maybe essential for it. May be the only antimicrobial that we can really safely take because it's not destructive to the good bugs. And so, what do we call this? I'd love to anybody to reach out or provide some brainstorming in Slack. Is it space milk? Is it galactic milk? Mars milk? Survival milk? I mean, how important do you want the name to explain? Astro milk. So I think just thinking about how to talk about human milk in a way that can go beyond the controversy of human milk on Earth is something that I think about, and I wonder if anybody has any thoughts on that. So yeah, that's it really. I think I believe in it, obviously. I've been pioneering mammary cell agriculture since 2013, and I'd love to continue the discussion with anybody, and happy to get to the questions now, Alex.
Great, thank you so much. And I think we have some questions coming in, and galactic milk definitely makes me think of the movie Spaceballs, and I think I can say that on here. But we have actually another, somebody says Milky Way milk. And so yeah, if you have questions, go ahead and put them in the chat. We'll start by taking a question from Scott, who says, you mentioned the microbiome of breast milk being critical. How can that be achieved with a cellular agriculture approach?
Well, the point is that your microbiome, an astronaut will take their microbiome with them, and they will not want to lose it. So if you do get sick, if there's something that gets out of balance in your microbiome, it could be fatal on Mars. It could be immediately fatal because before antibiotics, it often was. And then it could become fatal if you use antibiotics, because then you lose your microbiome, and how do you get it back? It's not really known how to reconstitute microbiome. I guess, there's stool transplant, and probably that can go a long way, but that can also go bad too. So how do you want to keep it in balance, and that's really the point. And there's nothing better for that than milk. In fact, there's nothing else that has ever worked other than milk and stool transplant, I would argue. And the reason for that comes from the list of molecules and the known methods of action in the human body, in vitro. These molecules have been deeply studied. There's just never been a supply of them. The HMO are really critical for feeding the good bacteria. Secretory IgA, which is early in 2020, I moved into antibody production and discovered a new way to make secretory antibodies, which are the antibodies that coat our entire mucosa throughout our entire lives, and they neutralize every viral and bacterial particle that we breathe in or that we eat. And that secretory IgA is antimicrobial and probiotic. They interact with commensal bacteria to maintain the microbiome. And then there's many proteins that have amazing properties. You know, I could talk for hours and hours about it, but there's nothing that compares to that list of molecules in human milk for suppressing the bad bugs and maintaining the good bugs in the microbiome. I'm not trying to say that you get a new microbiome when you drink biosynthetic milk, you know, because it's cell-free and it's pathogen-free. But it's about maintaining and never losing. You really don't want to lose your microbiome on Mars, and that there's probably nothing better than milk for that.
Great, yeah, and that really kind of opens up the can of worms, and really think about that. Another challenge we have to deal with in space. We have a couple more questions here. One is asking, can you explain more what you might need from bioreactor innovation?
Actually, the bioreactor innovation, you know, maybe larger, what they call cartridges. So bigger cartridges so that you have larger batches. So your bioprocessing is always going to be batch specific. So you're going to want to monitor each batch. Now, these are continuous production systems, secretory systems. So you set up a bioreactor and maybe it runs forever. Nobody actually knows. I think it's been about 18 months is the longest anybody's run these continuous secretory bioreactors. And it's really not even that innovative. It's media flow. It's, you know, the bioreactor is a combination of cells and hardware. So you know, there's still things to be discovered about how to maintain the level of certain nutrients and growth factors over time. And so automation, fluidics, you know, it's not really like an invention, but it's, you know, improvement and optimizing the cell production for this specific cell type. There's still many gains to be had in that way. And then automation, and this is actually a little trickier because there's plenty of science around how to process milk, but it's never been all milk ever made to date until, you know, January 20th, 2020, when we made, you know, we believe the world's first biosynthetic milk, had pathogens. It was not pathogen free. So you didn't have to think about how to do bioprocessing of ordinary milk because in a sterile environment. That's going to be on Earth and on Mars the primary obstacle, because again, these proteins and these molecules are so special. You really don't want to have to pasteurize them. So producing bioprocessing systems that are closed systems, automation, continuous monitoring, that's probably the biggest area of innovation. But even within that, you know, I think the technologies exist, and it's really just bringing these existing technologies and building that closed system. But you know, I don't think there's any sort of risk that you would look at and say, you know, it's possible this isn't going to work. I think we've kind of crossed that threshold and we've said, okay, we have engineering challenges. Those are primarily capital challenges. Can we hire enough engineers to solve these problems? And I think it'll happen.
Great. We have two questions here that we're going to kind of bring together. One of them is, you know, what is the starting point for the milk producing cell cultures? Do you cultivate primary mammary epithelial, or do you transdifferentiate it from other more accessible autologous adult cells? And then the follow-up is, have you tried using mammary gland epithelial cell mammospheres or organoids?
No, it's all primary cells and immortal cell lines right now. We also do some stem cell. I know some competitors are focused on stem cells. We've found that primary cells work quite well, and you want there's it's up for debate how many mammary phenotypes there are actually out in the world based on, single cell sequencing. Some people think there's 16 different types of cells in the mammary gland that contribute to milk production. So it works with primary. I think it works with stem cells, but I haven't actually proven that personally. Immortal is not as good as primary, so it's kind of interesting. We go back to this sort of cannibalistic thought, which is even if all your cell lines die, every astronaut has the cells inside of them. My focus is on primary. I'm really trying to maintain that non-genetically modified path. With food grade media, primary cells producing the most nutritious and therapeutic drink that can sustain human life. So, that's my sort of vision and path. But I think that, given enough research and investment, probably all of those are viable. And like I said, I don't want to reinvent the cell, so I stay, focused on cells.
Chris is asking, how many calories do you get from 0.75 liters? Will there be additional, or where will the additional calories come from?
I wish I should know that number off the top of my head. You could make enough milk, I believe, to sustain an astronaut, but it would be more than 0.75. I think you're getting somewhere like 400 to 500 calories, maybe at 0.75 liters off the top of my head. I really should know that number. I apologize. But the point is more that, the amount of mass that we would need to ship to Mars is feasible. That, it's not just for milk. A lot of those amino acids are going to be useful for other cell agriculture. And I think milk is just kind of unique from an energy and sort of a resource perspective. There's a fundamental problem with meat. You put all this effort into growing your cells, and then you eat them. Like that, like they're gone. But you grow these mammary cells, you get them happy, and they start making milk, and they might make milk forever. And so the potential is there. Certainly in an emergency situation, you should be able to drink, more than your supply of daily milk that is typically rationed, I'm assuming, on Mars. If your crops fail and you need to ramp up your milk production, that's probably sufficient to sustain life on Mars. But yeah, I think it's more about those molecules are really special for maintaining your microbiome. And so you want to have consistent access to those molecules because, you drink it, it moves through your system, and you want to continue to kind of, feed your microbiome. So, that it's not the only solution. It could be the only solution. But, who's going to want to just drink milk every day for the rest of their lives? Maybe on Mars, the people who are going to be down for that are going to be ready for those kind of environments. But I think it's really more about just maintaining the overall health, and it's a clear substance that can be part of the feeding picture for maximum, health and maintenance and prevention of, pathogen infection, which, can get really dangerous on Mars.
Great. So we have a couple additional questions, some related to antibodies, some related to FDA approvals. We will go ahead and move these over to the questions for answers Slack channel. Shayne, thank you so much for the presentation.